this lesson, you will see how a student follows the scientific method. The first step in performing any scientific experiment is asking a question. In this case, we are going to investigate whether or not red cabbage juice is an indicator. An indicator is a chemical substance that changes in a predictable way when combined with another substance. So we want to know if red cabbage juice can indicate the pH of a solution, whether it's acidic or basic. Once we have our question, the next thing we need to do is form a hypothesis. The hypothesis is an answer to the question, a possible answer to the question, and it creates a prediction for what you expect to see in your experiment. It is written in an if-then format. So we will say, if red cabbage juice is an indicator, then it will change color when different substances are added. Watch as the student conducts the experiment, following a specific procedure, and then afterwards we can analyze the results. In order to make red cabbage juice, 1 cup of water was brought to a boil on a stovetop. Once the water was boiling, a few cabbage leaves were added to the water. This was allowed to sit for a few minutes. Once the red cabbage juice was created, it was poured off into a measuring glass. You can see it has a purpley color. 15 drops of red cabbage juice were added to each of 3 test tubes. Some of the juice was retained to serve as comparison. ½ teaspoon of baking soda was added to a cup. Enough water to dissolve that ½ teaspoon of baking soda was added to make a clear solution. It would be added to the purple cabbage juice. 10 drops of the baking soda solution were added to one of the test tubes containing the purple cabbage juice. You can see that the color has changed. If we compare it to our control, you can see how the original color compares to the color after adding the baking soda. 10 drops of vinegar were also added to another test tube to see the color change there. You can see we're getting a red color when adding vinegar. We also used 10 drops of ammonia and added that to the red cabbage juice. This produced a blue-green color. Here you can see the results of the experiment. When we compare them to the control, the cabbage juice with nothing in it, you can see the difference in color from the original cabbage juice to the cabbage juice with these different solutions added to them. The results of the experiment were recorded in this data table, which we titled "The Effect of Chemicals on the Color of Red Cabbage Juice." We had 4 different test tubes. The starting color of the cabbage juice was a reddish purpley color. We can see the substance added and the final color. Test tube 1, we added ammonia. The final color was a blue-green. In test tube 2, we added vinegar. The final color was red. In test tube 3, we added baking soda. The final color was also a blue-green. And in our control test tube, in which we did not add anything, the final color was the same, that purpley red color. If we assume that red indicates that a substance is an acid, and that blue or green indicates that the substance is a base, then we can see that vinegar is an acid, and ammonia and baking soda are both bases. Based on this information, we can conclude that cabbage juice can be used as an indicator for pH.